what I'm going to be doing today is taking you through some of the information that was presented in the Fascia Congress in 2007, and some of it that was presented in 2009. And I'm going to give you opportunities to ask questions and summarize some of the findings, and then tell you what I think it means to me as a clinician and find out from you what you might like to see. What is fascia? Well, Gray's Anatomy defines it as fibroareolar or aponeurotic laminae of variable thickness and strength found in all regions of the body. It covers every structure in the body: bones, muscles, nerves, blood vessels, organs. It lubricates, it insulates, it transmits force. So fascia is what we're going to be talking about. Fascia is what concerns us as clinicians. If you then have a cell here, and you have a micropipette, and you push on the membrane of the cell, here's the nucleus. When you push on the membrane, the nucleus suddenly puffs up. You can see this in the living cell. Then again, mechanical forces on the cell membrane instantly affect the cell nucleus. The cells are primed to pay attention to their mechanical environment.
Do you have any hypothesis why the nucleus puffed up?
The question was, I showed the slide of when you push on the outside of the surface, the nucleus puffs up. The question is, why does the nucleus puff up? The nucleus is a compact, the DNA is tightly coiled. If you're going to do something with a tightly coiled DNA, you've got to uncoil it so you can get at it. So the nucleus has to become larger so you can then transcribe the DNA. So the nucleus puffs up a little bit, the DNA gets transcribed, the RNA starts coming out. So there are clearly mechanisms by which the mechanical environment affects the cell. Whether that means that when we do something, we're actually affecting the cell is a different question entirely. But if you back up a little bit and you think of the embryology, all the information required to develop this whole body is packed into one cell. Those cells divide. By the time there are about 16 cells, they're still identical, but they're starting to change. So now you have 16 identical cells, any one of which could form a human. And that's when they split, you get identical twins. At that point, the mechanical environment is the same because it's a round. It's a torus, it's a round structure, and all the cells are the same. But once they get more than 16, the mechanical environment in the inside is different from the mechanical environment on the outside. And so the cells on the inside start developing differently. And as they develop differently, then there's a different mechanical and biochemical environment, and so the whole structure gradually unfolds. By the time we become adults, we can't take individual cells and create new adults from them. In other words, the cell has gone down a path and it can't back up. But the mechanical structure affects, again, even within the adult tissue, the mechanical structure does affect how those cells do respond.